All right, welcome back. I hope everyone had a nice lunch and some time to socialize with colleagues. I hope some were able to attend the Mercy Ships lunch, professional coaching session, or the SOAPS town hall. I'm Hester Shea, and I'm one of your moderators today. I'm an assistant professor of surgery at Johns Hopkins All Children's Hospital. My co-moderator is Dr. Stuart Carter, and he is an assistant professor of surgery at Norton Children's Hospital. This session focuses on necrotizing enterocolitis. It's my honor to introduce our first presenter. Dr. Colton Wayne, a pediatric surgery research fellow at Nationwide Children's Hospital, and he'll be speaking on the association of socioeconomic status with development and severity of necrotizing enterocolitis.
Thank you for that introduction. Good afternoon, my name is Colton Wayne. I'm a second-year surgical research fellow at Nationwide Children's Hospital. Thank you for the opportunity to present our work. We have no disclosures. The purpose of this study was to determine if there are important associations between socioeconomic status and necrotizing enterocolitis, or NEC. This research is important as any significant associations identified can potentially alter maternal risk assessment and drive parental guidance to improve patient outcomes for NEC. Known risk factors for NEC include prematurity, formula feeding, and hypoxia. Dysbiosis of the gut microbiome may also play a role. Social factors linked to NEC development include Hispanic ethnicity and non-Hispanic Black race. A recent study by the Markle Group found that social determinants of health, or SDH, are linked to mortality in NEC. In contrast, other previous studies showed that insurance status and median household income, which are considered markers of SDH, were not predictive of mortality. This highlights that there is uncertainty about the impact of SDH in cases of NEC. Prematurity, the greatest risk factor for NEC, has been linked to low maternal and familial socioeconomic status, or SES. Women of a lower socioeconomic status have increased difficulty providing breast milk, which has known protective factors against NEC. Additionally, lower socioeconomic status is associated with alterations of the adult gut microbiome, which has a direct impact on the microbiome in infants. Aside from prematurity, it is unclear whether socioeconomic status is an independent risk factor with the development of NEC. As clinicians and researchers, we must ask ourselves what can be done to prevent the development of NEC and how we might predict its occurrence in advance of clinical disease. With this in mind, the aims of our study were to determine whether neonates of a lower socioeconomic status have a higher risk of developing NEC, and to assess whether neonates of a lower socioeconomic status are more likely to develop severe NEC requiring surgical intervention. The Pediatric Health Information System, or PHYS database, was queried for infants 60 days of life or less with a diagnosis of NEC from 2013 through 2023. Cohort and case control studies were conducted to determine the association between NEC and SES. In the case control study, infants were one-to-one matched as infants with NEC and healthy controls based on gestational age, chronological age, race and ethnicity, and the presence of cardiac disease, and the need for surgery was identified using ICD-10 procedural codes. Socioeconomic status was measured independently using either median household income based on zip code or child opportunity index scores, and statistical analyses were performed using multivariable logistic models to measure associations of these metrics with the development and severity of NEC. Additional mediation analysis was performed to assess the extent of the association between NEC and low SES that was mediated by prematurity. Child Opportunity Index uses patient zip code information and neighborhood factors, focusing on access to education, health and environment, and social and economic factors. With these, a composite score is generated, placing children into very low, low, moderate, high, or very high opportunity index. We identified 11,151 infants with NEC in the PHYS database, and after one-to-one matching, our sample group consisted of 11,100 infants with NEC and healthy age-matched controls of the same number. With our demographics, 57.1% of infants with NEC were male, 38.1% were of non-Hispanic white race, and 39.1% had an extremely low birth weight of less than 1,000 grams. The primary source of health coverage for our patients with NEC, managed either medically or surgically, was government insurance at 64% and 65% respectively. In the cohort study, compared to patients without NEC, patients with NEC were significantly more likely to have lower socioeconomic status as measured by median household incomes in the first to third quintiles, and very low, low, or moderate child opportunity index. These findings were confirmed in the case control study, in which patients with NEC were significantly more likely to belong to lower median household incomes and child opportunity categories. Importantly, mediation analysis based on MHI or COI revealed that only 29.2 and 27.5% of these associations were mediated by prematurity. Next, we assessed factors associated with the need for surgical intervention for NEC. In an adjusted model, neither MHI nor COI were associated with requiring surgery. Our study was limited by its retrospective nature, and additional limitations are those inherent to large database studies and are shown here. Regarding future directions for this project, we plan to perform a local study at Nationwide Children's Hospital using patients with NEC and age-matched controls to assess SES impact and the development and severity of NEC. And access to their electronic patient records will allow us to ascertain greater detail about patient characteristics, hospital course, census tract level child opportunity index data, and long-term outcomes for these infants with NEC. In conclusion, among infants of the same level of prematurity and race and ethnicity, lower SES is an important factor for the development of NEC. Notably, less than 30% of this association was mediated by prematurity, which suggests that other factors such as the gut microbiome or availability of breast milk play an important role for the remainder of that association. Targeted interventions to support low socioeconomic status families are important and may reduce the risk and disease burden of NEC in these situations. I'd like to thank my mentors and collaborators for their help with this work and all of you for your attention, and I'd be happy to take any questions.
Christian Manhart, Indiana University, great talk, and something that I think a lot of us are looking into. One thing that we've been looking into at Indiana is the breast milk conundrum, and notice with the fact that there's lower socioeconomic status, there is a lower availability of breast milk. And so I wonder if you had looked into or had thoughts of why we have kind of lower availability of breast milk, because we've seen both on the maternal end, there's discrepancies in what they view of like, is breast milk really necessary versus formula feeding? And also there's obviously a decrease in availability to breast milk with milk banks and being in a NICU far away from home. So any thoughts on any of that?
Yeah, that's a really great question and point, and it's something that unfortunately in a FIS database study, you can't get granular information like that, and that's part of what I'm hoping to do with our local study, is be able to look into some of the factors that may impact availability of maternal breast milk. I know that at Nationwide Children's, there's constantly information being disseminated to mothers of babies in the NICU about the breast milk program, whether it's donation or receiving such breast milk. And I think that looking at a larger scale, because what we're trying to do is identify things that can be acted on before a baby is born, is working with OBGYN colleagues, and if they can identify mothers that are at risk in the situation that we've identified, maybe connecting them with those resources early.
Thank you.
In regards to your geographic information, was there any ability to, compare lower socioeconomic status and see if there's any patterns based on geography, access to resources, environmental toxins, et cetera, that might have predisposed mom to having issues?
Yeah, that's a great question, and I do believe that as part of our initial data set, we gathered information on regions based on what's available in FIS. As part of a follow-up, before the local study, we're also doing a follow-up with the FIS data to look at outcomes, and specifically we can also focus on some of that in that, so that's a great idea, thank you. All right, thank you, everyone.
Thank you. Our next presentation is impact of early versus late enteral feeding reinitiation on tissue regeneration after necrotizing enterocolitis in mice, a basic science approach. Dr. Lena Bode, postdoctoral research fellow, Johns Hopkins Children's Center.
Good afternoon. In this project, we approach the initiation of enteral feed after NEC from a histological perspective. I have no disclosures. The reintroduction of feeds is an ongoing discussion, training the bowel without overwhelming the still injured intestinal epithelium. We examined how to find a safe time point to examine intestinal tissue regeneration. In our murine neck model, we induce NEC in 7-day-old pups, which we feed with formula. This contains bacteria we isolated from a human neck patient and expose these mice to hypoxia 2 times a day. We think it's important because feeding strategies can influence intestinal tissue regeneration. And although it's a murine neck model, we hope to get more information about risks and benefits of early feeding strategies and how to take this into our clinical decision making. I'd like to explain you our strategy of those 2 feeding schemes. The mice were exposed to the experimental neck for 2 days, and after that, they were split up into 2 groups. The early feeding group had only a short period of 5% glucose, feeds after quickly accelerating to full enteral feeds of plain formula for 42 hours. This means we challenge the bowel right away with complex digestive processes. The late group had a full day of 5% glucose, a mild stimulation for the bowel, taking up the glucose with SGLT1, and then water follows osmotically driven, and then a full day of formula feeds before sacking. Let's get to the results. We could see that the early feeding kept the mice in a stable body weight. Furthermore, we assessed histomorphological features. We looked into the villi length and the crypt area. To the very left, you could see healthy age-matched breast fit controls with the black dots, and then we showed that the early feeding initiation group had longer villi and enlarged crypt areas, significantly increased compared to the late feeding group. Then we assessed motility. We did that on day 5 by feeding a fluorescent dye to the mice and assessed how far it traveled down the intestinum after 30 minutes. On the right graph, you can see the black curve with the healthy breastfed mice where the dye reaches furthest, and NEC and early and late feeding groups were behind. The left graph indicates how much of the dye, what amount is in the small intestinum. In acute NEC, the far right group has the lowest amount of the dye. The dye amount is increased in the late and early feeding initiation groups, and then best group is the best fed group. As we already saw the increased and enlarged crypt area in the early feeding group, we assessed proliferation markers by Ki-67 and saw that the early feeding initiation enhanced crypt proliferation significantly. Moreover, it's important for tissue recovery to evaluate epithelial integrity. The top panel is the early, the lower panel is the late feeding group. Both feeding schedules reached similar epithelial integrity markers. We assessed E-cadherin and ZO1, anti-epithelial tight junctions protein, and the beta-catenin that anchors the ECAD to the actin skeleton of the cell membrane. Then we took a look into inflammation markers. We did a multiplex ELISA on the plasma and checked the cytokines. And it was interesting that the early feeding group had significantly higher macrophage attracting cytokines. And we wanted to validate these findings and performed flow cytometry to check how the actual macrophage populations are in the lamina propria. Both groups showed similar amounts of macrophages, and within these macrophage populations, early and late feeding had predominantly M2 polarized macrophages, so the anti-inflammatory recovery type. To summarize our histological findings, the early feeding initiation revealed a higher proliferating rate. Both early and late enteral feeding had comparable motility and epithelial integrity markers and similar amount of anti-inflammatory recovery macrophage populations. Thank you.
Hey, nice presentation. Thanks for bringing that to us. Andrew Zigman, Portland, Oregon. You know, we get scared in our human mice to feed them too early after they recover from necrotizing enterocolitis. We're afraid about the tonicity of the milk and whatnot. Can you tell us a little bit about the formula or the milk that you gave your experimental subjects?
Yeah, so the formula we fed is just a normal formula that you would give to babies. So we don't work with a certain company. It's just the plain milk formula.
So do you know, for example, if it had, so it was an artificial formula, not milk, and do you know, for example, the tonicity of it, the osmolarity?
No, I cannot say that the osmolarity of that formula.
Okay, well, maybe this will make us less scared of refeeding our babies afterwards. Thank you.
Gail Besner from Columbus, Ohio. Thank you for your talk. I may have misunderstood this, but it looks like before you randomize your animals to the early and late feedings, you induce NEC for 2 days. And in general, I think that the HACM lab and our lab for sure find that it takes more time to really induce NEC, and maybe by day 2, the animals might just be starting to get sick, but we typically give them stress for a longer period of time. So I'm wondering if you did that and whether it would change your results. Thank you.
Thank you for your question. So our NEC model differ between 2-day NEC models to 4-day NEC models. Sometimes we check especially epithelial injury after 4 days, but in a time course we could already show that our cytokine levels peaked at day 2. So we took that time point with the peak cytokine levels, and from then on started the different feeding regimens.
Thank you. Okay, our next presentation is argon inhalation promotes M1, M2 macrophage polarization during experimental necrotizing enterocolitis, presented by Felicia Belasmo, a graduate research student at the Hospital for Sick Children.
Good afternoon, everyone. Thank you for allowing the opportunity to present our work with you here today. I'm Felicia Balsamo from the Hospital for Sick Children in Toronto. Now, neonates with NEC are frequently mechanically ventilated, and based on our recent analysis of NICUs in Toronto, as well as our international multi-center prospective randomized control trial, which we coordinated, we found that 83 to 89% of neonates with NEC are mechanically ventilated, which shows that delivery of inhalational compounds in the respiratory circuit of ventilated neonates with NEC is practical and it can be achieved clinically. Now, noble gases are chemically inert gases that have gained considerable amount of attention due to their clinically useful biological effects. And inhalational therapy does not only treat lungs, but can also extend to systemic treatment of many other organs. So this is why it's beneficial. Argon is the most abundant noble gas and has been of increased interest due to its low costs, non-toxicity, and excellent safety record, both in vivo and in adult human studies. The beneficial effects of argon have been shown in multiple organ systems, and mechanistically, argon has been shown to attenuate inflammation and apoptosis via TLR4 signaling. However, argon inhalation, the mechanism of action, has not been explored thus yet in a neonate or in experimental NEC. And we have recently shown, last year at APSA, that argon inhalation reverses NEC-induced intestinal injury. So mice with a histological score or grade of 2 or higher were considered to have NEC, and argon inhalation had a significant reduction in these NEC grades. as well as it attenuated intestinal inflammation shown by decreased gene expression of TNF-alpha and IL-1/6. So this diagram illustrates the polarization of macrophages from the naive M0 state into either M1 or M2 phenotypes in response to specific stimuli. So upon recognition of LPS by TLR4 signaling, M0 macrophages differentiate into M1 macrophages, which are pro-inflammatory, and they can secrete cytokines like TNF-alpha and IL-6, and they express the cell surface marker CD86. In contrast, we have M2 macrophages, which are activated by IL-4 and IL-10, and they are anti-inflammatory, as well as they promote wound healing, and they express the cell surface marker CD206. Now, during NEC, polarization of M1 pro-inflammatory macrophages contributes to NEC development by enhancing inflammation via TLR4 signaling and lytic cell death in intestinal epithelial cells. However, argon has been shown to actually regulate M1 towards M2 polarization in macrophages. So the aim of our study is to investigate whether argon inhalation can alter the M1 to M2 macrophage balance during experimental NEC. Our NEC model was induced from postnatal day 5 through postnatal day 9 through gavage feeding of a hyperosmolar formula and LPS, as well as intermittent hypoxia. In the NEC and argon group, NEC pups received argon at a concentration of 70% argon, 30% oxygen. And why we chose this concentration is because many in vivo studies use this dose, as well as a safety study in human adults use this 70-30 composition. So mice were placed within a sealable chamber connected to this gas tank composed of 70% argon, 30% oxygen, all placed within an incubator from P6 to P8. Now, differential protein expression analysis in intestinal tissue lysates showed that argon reduced pro-inflammatory TLR4 signaling as well as cell death, shown by reduced expression in blue color, rescuing the NEC-induced intestinal damage as shown in my previous slide. This also supports our hypothesis that M1 pro-inflammatory phenotype is decreased. And further, we found proteins signifying M1 macrophage polarization were decreased, while those of M2 macrophage polarization were increased following the administration of argon. Therefore, argon alters the balance of M1, M2 macrophages towards that anti-inflammatory M2 phenotype. Further, we conducted immunofluorescence staining on NEC intestinal tissue from all groups and stained for M1 markers CD86 as well as M2 markers CD206. And we found that the ratio of M1 to M2 activated macrophages was significantly increased in the NEC group compared to the control and subsequently reversed following the administration of argon. So in conclusion, argon inhalation during experimental NEC reduces intestinal injury by inhibition of TLR4 signaling and cell death, and also alters the balance of M1, M2 macrophages towards that anti-inflammatory M2 phenotype. Now, the translational potential for argon is great as it is a novel and practical therapeutic intervention, as neonates with NEC are frequently mechanically ventilated. I would like to thank all members of the Puro Lab as well as our funding agencies, and I welcome any questions at this time. Thank you.
I know very little about argon inhalation, but I was curious. My impression was that being exposed to argon gas wasn't necessarily a good thing in the environment, but is there a difference in the composition from what a natural environmental gas leak would be, or something in the industry versus inhalation?
Yeah, so the composition of argon in our atmospheric air, I believe, is 1%. And in terms of what it's used in the industry, I'm not too sure of. In the literature, there's many concentrations of use ranging from 20% all the way up till 80%. So there is quite a vast range of the concentration that's used in terms of argon, also ratio to oxygen.
There was also a few studies that were mentioning neuroprotective nature of argon inhalation, and I was just wondering if I know this is a NEC-focused study, but considering the frequency of brain-related issues in that population, whether that would be a metric to look at in the future.
Yes, of course. We do know that there's a lot of neurodevelopmental delay in these neonates, these NEC survivors, and we've already taken a next step towards investigating the neuroprotective properties of argon in our NEC model, and that's one thing that we're going to expand on as well. We found that it is also protective. It decreases activation of microglial cells, so in conjunction with our macrophage story in this presentation here. So we're hoping to expand on that in the future as well.
Nice work.
Thank you so much.
Our next presenter is Dr. Cody, forgive me if I mispronounce this, Gesser, and he's going to be up here for 2 consecutive presentations. The first of which is administration of specific human milk, oligosaccharides protect against enteric glial loss and small intestine hypomotility in experimental necrotizing enterocolitis.
Okay. Thanks so much. No disclosures. So necrotizing enterocolitis is a devastating gastrointestinal disease of premature infants. Our lab and others have demonstrated a critical role for disordered TLR4 signaling on the intestinal epithelium in NEC. And I don't have to convince anyone in this room, small intestinal dysmotility is one of the earliest signs of NEC and often precedes its fulminant clinical presentation. We have abdomens that look like this and abdominal X-rays that look like this. So hold that thought. Human milk oligosaccharides, or HMOs as I'll refer to them, which are non-digestible carbohydrate components found in human breast milk, have been shown to be protective against experimental NEC, but by incompletely characterized pathways. So we hypothesize that HMOs can reduce the risk of experimental NEC by restoring intestinal motility. We tested this hypothesis in our well-established murine NEC model that's been described a few times already, so I won't go into much more detail. Just to say that the mice that were treated with NEC plus the HMO treatment had either the compound 2-fucosyl lactose, or 2FL, or 6-sialyl lactose, 6SL, or a blend of these. So there were 3 formulations of HMO that we used. And then just a word on the FITC dextran motility assay, because it's always a little hard to wrap your mind around the first time you hear about it. The mice are orally gavaged with FITC dextran exactly 30 minutes before sacrifice. And then the stomach and small bowel are segmented into 1 centimeter pieces, and the FITC content within each centimeter of small intestine is read on a plate reader from a homogenized sample. So first, just to establish that HMOs protect against NEC development, we looked at first histologic injury on H&E stained slides. The slides were all rated by blinded observers, and their scores are indicated here in the lower left-hand panel. We also looked at the expression of lipocalin-2, which is a marker of mucosal inflammation in mice. And then we stained for the nitrative stress marker 3-nitrotyrosine, which is quantified in mean fluorescence here in the lower right-hand panel. So you can see that histologic injury, lipocalin-2 expression, and nitrative stress are all induced in this NEC model, and then all mitigated by the administration of HMOs independent of a specific formulation. Then we looked at motility. So this is the graph, again, of the mean FITC fluorescence at each centimeter of bowel for each group. And then over here on the, oh, you can't see my pointer. The far right is 2 graphs of geometric center of the FITC bolus for each mouse. On the top is healthy controls, on the bottom are the NEC mice. So you can see that at baseline, these compounds don't influence intestinal motility when supplemented, but they do rescue NEC-induced hypomotility, again, regardless of their specific formulation. Then we looked at the effect of the HMOs on the density of enteric glia in the tissue, and we stained for the marker SOX10 of enteric glia. The insets you can see down here in the second row, and the quantification of the glia is the mean density of SOX10 fluorescence. You can see there is NEC-induced enteric glial loss in the model, which is partially restored with all 3 formulations of HMOs. And then lastly, to begin to get at the mechanism, we examined the effect of the HMO treatment on TLR4 signaling in an in vitro LPS stress experiment of cultured enteroclia. So the groups that got HMO were pretreated for 30 minutes, and then all the groups were exposed to LPS for 45 minutes, after which the cells were all fixed and stained for the NF-kappa B P65 subunit. To help understand these images, here's just a quick review of the canonical TLR4 pathway. Starts with LPS engagement of TLR4 at the plasma membrane, and then ends in NF-kappa B translocation to the nucleus. So if we can visualize NF-kappa B green signal in the images, we only quantify the signal which co-localizes with the blue DAPI, the nucleus. So you can see here quantified on the far right, there's induction of NF-kappa B translocation in the LPS exposed group, which is all knocked down with HMO pretreatment. So in summary, the HMOs were protective against NEC development in an experimental model. They rescued NEC-induced small intestinal dysmotility. They rescued NEC-induced enteric glial cell loss, and in an in vitro model, they reduced TLR4 signaling in enteroclia. So, attenuation of enteric glial cell loss and the subsequent hypomotility may represent just one of the many protective pathways that these compounds offer. Thank you, and I'll take any questions on that presentation.
Thank you for the talks. I have a question regarding the glia cells and from HMO. So you all gavage the HMO to the GI tract. So the HMO has to pass the epithelium, pass the muscles, and reach the neurons. So how was the interaction between the HMO to the glia cells?
That's a great question. They are, I think, internalized. It's just not a digested complex carbohydrate for metabolism, but they are able to cross the enterocyte and then enter the enteroglial cells in the lamina propria and in the muscularis.
Is it possible to interact with the EC cells, not directly to the neurons, but also to the EC cells, which also has the role to regulate the gastro motilities?
The enteroendocrine cells?
Yeah.
Certainly, yeah. Yeah, there are receptors for those sugars on them as well. Yeah. Thank you.
What was the, and I might have missed this, the rationale for 3 different types of oligosaccharides, and was there any kind of expected difference that you didn't notice?
Yeah, I think it was more to understand whether it was an individual compound or a class effect, which is why there were 3 different types used as well as a blend, which clearly it showed it was a class effect. Yeah.
All right, thank you.
Thanks.
All right, and your next presentation, plasma TLR4 level.
Thanks, everybody.
Plasma TLR4 levels as a biomarker of intestinal TLR4 activation in a mouse model of lipopolysaccharide endotoxemia.
All right. So again, no disclosures. So broadly speaking, pediatric sepsis is a major cause of morbidity and mortality in pretty much any disease state in children. To reduce progression to septic shock and multi-organ failure, early diagnosis and treatment are of course critical, and to that end, early biomarkers of sepsis are sought after. And of course, we're going to talk about this in terms of NEC, which is a very common and very devastating cause of abdominal sepsis in preterm infants. Early NEC represents the window of reversible intestinal injury, but of course, it's notoriously really hard to diagnose. Having spent a lot of time demonstrating the role of the toll-like receptor 4 in the pathogenesis of NEC, we therefore hypothesized that the toll-like receptor 4 itself could serve as a detectable biomarker of early NEC specifically. So we first conducted an LPS challenge experiment in adult mice as proof of concept of the assay and also to validate the specificity of our ELISA for the protein. We treated either C57 Black 6 wild-type mice or TLR4 global knockout mice with either saline or LPS intraperitoneal. And after 6 hours, sacked whole blood as well as distal ileum. The whole blood was centrifuged. Plasma was isolated with great care taken to avoid the buffy coat, and then assayed for free TLR4 using a sandwich ELISA assay. And then the intestinal sample was processed for ileum qPCR inflammatory cytokines. So this is the plasma TLR4. You can see that in the wild-type LPS-treated mice, there is a clear and robust secretion of TLR4 into the plasma, a change which is blunted in the TLR4 knockout mice, validating the specificity of this assay for the protein. And this also corresponds with the intestinal inflammatory cytokine profile that we see in the same mice. So then we took this assay and applied it to a NEC model. Again, the same NEC model that my colleagues have described. NEC was induced, and the mice were sacked quite early at various time points in the model. It's pretty small text, so you can't see, but this is 6 hours, 12 hours, 24, or 48 hours after the first oral gavage of the NEC formula. Again, the blood was processed and the plasma was assayed for the TLR4 protein, just as before, but also with the cytokine multiplex ELISA to examine the secretion of other pro-inflammatory cytokines. So here's the plasma TLR4 in the NEC model. You can see from the baseline control value, there is an abrupt and early increase 6 hours after the first gavage of the NEC formula, which then declines steadily until it's at or just below its baseline value at around 48 hours. And the cytokines from the multiplex assay follow a very similar trajectory. Here's IL-6, MCP1, and IL-12. So lastly, we use this assay in another time course model, but instead of looking at the plasma, we looked at the stool. The mice were sacked at either 24 or 48 hours. The stool was rinsed from the entire length of the colon. It was homogenized in a protease inhibitor cocktail. It was assayed for total protein on a BCA assay, and then the samples were assayed for TLR4 using the same sandwich ELISA. And you can see from a baseline level of essentially undetectability, there is an abrupt rise in stool TLR4 on day 1 of the NEC exposure, which continues into day 2. So in summary, we demonstrated administration of LPS to wild-type adult mice leads to a rapid and robust TLR4 secretion through a specific TLR4 ELISA kit. This carries over to a NEC model, which demonstrates a rapid and early robust rise in TLR4 early in the model, and that it's detectable in the stool above an undetectable baseline early in the model as well. We think this could help with early diagnosis of NEC in pediatric sepsis. Thank you.
Agostino Piero, Toronto. Thank you very much. Very interesting paper, and congratulations for your group for doing these excellent studies. I wanted to ask you, I mean, one of the main problems is distinguishing a baby with NEC from a baby with a sepsis. Now, and not all the babies with sepsis have NEC. So do you think that these tests will actually have a lot of confounding factors, like generalized septicemia, which may not be able to help us in the early diagnosis?
What was the, like what? Which, I couldn't hear the
like
final word.
measuring TLR4.
Yes, I mean, yes and no. So this is generally probably just a marker of gram-negative bacterial activation of an innate immune pathway, which is the pathway that we've shown is critical for NEC, but is of course involved in numerous other biological processes. So gram-negative pneumonia, of course, you may see plasma TLR4 rise, and you may even see it excreted in the stool. I don't know, we haven't looked at that. But non-NEC infections with the cognate ligands to TLR4 could of course serve as confounding factors in this clinically, which is why it would never function as a standalone test, of course, but would require clinical context.
Okay, thank you very much.
Thank you.
Say this makes its way to the clinical side. The assay for TLR4, is that something that would be problematic for widespread institution, for labs and hospital systems? And then in terms of looking at fecal TLR4, is there some way you can identify that at bedside without having to send specimens?
Yeah, I mean, that's Those are two questions that we've contemplated in developing this. So the first being, would it be difficult to implement? The answer, I think, is no. This was a commercially available ELISA kit, which could be developed for a clinical scale quite easily with the proper partners, which I think segues nicely into your second question, which is, could this be a bedside test? And I think I would venture to say possibly yes, just given the binary nature of what we saw in our first fecal TLR4 assay, which was essentially a 0 to a something elevation. So yeah, a qualitative bedside test I could absolutely see this emerging into.
All right, thank you.
Thank you.
Our next presentation is shorter antibiotic duration and stage 2 necrotizing enterocolitis does not increase stricture rate by Dr. Carlos Huerta, general surgery resident at University of Miami.
Thank you for the opportunity to present our work today. We have no disclosures. As extensively discussed today, necrotizing enterocolitis is characterized as a multifactorial inflammatory condition affecting neonatal intestine and further characterized by ischemia and dysbiosis of the neonatal gut. While the foundational principles of management, including antibiotics and bowel rest, have been well described and recognized, specifics with regards to the optimal duration of therapy for antimicrobial coverage remain to be highly debated among individual institutions and even providers. Treatment severity can be further characterized, as we well know, with the Bell staging classification system, for which stage 2 NEC can be associated with radiographic criteria, including pneumatosis intestinalis and portal venous gas. These types of radiographic criteria can be further portend intestinal complications during the same hospitalization for an infant, including intestinal strictures, which are a well-recognized complication of necrotizing enterocolitis and can occur in up to 25% of affected infants. As a result, we sought to understand whether the implementation of a 7-day protocolized antibiotic regimen would be associated with any differences in overall stricture formation among the same hospitalization for neonates compared to longer antibiotic courses for those with stage 2 NEC found on abdominal radiograph to include portal venous gas or pneumatosis intestinalis. We initiated a prospective institutional data registry within our institution in October 2015, and in August 2018 implemented a 7-day protocol of antibiotics including IV oxacillin, tobramycin, and metronidazole across our NICU. Data accrual for this study continued through December 2022 to ensure an equal and accurate number of comparator cohort for which to stratify patients into those who were treated prior to and after the implementation of this protocol. The month of August was also included and not included in this study as a washout period for the implementation of this protocol during our NICU. Our primary outcome of interest for this study was the formation of intestinal stricture, and secondary outcomes included overall antibiotic duration, protocol adherence, and progression to Bell stage 3 necrotizing enterocolitis. Among our overall study, there were 67 neonates identified with stage 2 necrotizing enterocolitis, of whom 49% were treated prior to the implementation of this protocol, and about half were treated afterwards. Among protocol adherents in those treated after 2018, this was described in 22 neonates, or 65% of this group. The most common reasons for protocol non-adherence was the presence of concomitant infections for which antibiotic duration was extended by the NICU intensivists, which occurred in about 3/4 of those patients who did not adhere to the protocol. There was a predilection for female sex in this cohort at about 55%, and these patients had a mean gestational age of about 29.7 weeks with an average birth weight of about 1,400 grams. The overall rate of intestinal stricture in this patient population was 9%, and there were 6 neonates who progressed to stage 3 necrotizing enterocolitis, or about 9%. Importantly, of those patients who progressed to stage 3, all cases occurred within the first 3 days of antibiotic therapy, with no patients that progressed to stage 3 NEC after cessation of antibiotics in any treatment arm. When comparing those patients treated prior to and after the implementation of this protocol, there were no statistically significant differences in many baseline characteristics, including female sex, gestational age, birth weight, the day of life for next stage 2 diagnosis, and the rate of intestinal stricture formation was similar in range from 7 to 9% respectively prior to and after the implementation of this protocol. Overall mortality and length of stay were similarly not statistically significantly different between both groups. The median duration of antibiotic therapy for those treated prior to 2018 in our study was 11 days, with an interquartile range of about 10 to 14 days. When excluding those patients who were non-adherent to this protocol, which resulted in about the after arm being about 22 patients, there were similarly still no differences in either female sex or gestational age. This group of patients had a somewhat lower average birth weight at about 1,100 grams compared to 1,700 grams in those patients treated prior to 2018. And once again, there was no difference in the day of life for next stage 2 diagnosis or intestinal stricture rate, which was again about 10% in this group. Overall mortality and length of stay were similarly no different between both groups. Ultimately, these results suggest that neonates with Bell's stage 2 necrotizing enterocolitis may be able to benefit from a protocolized 7-day antibiotic regimen without any associated increased risk of some intestinal complications such as stricture formation compared to neonates who receive longer courses of antibiotics. Furthermore, within this study, no cases of progression to stage 3 were noted after the cessation of antibiotics. And taken together, these results may suggest that newborns may be able to be exposed to shorter antibiotic courses, and future studies should continue to attempt to delineate the optimal and shortest course of antibiotics possible for patients with this disease. I'd like to thank all of my co-authors on the project, particularly my lead author and mentor, Dr. Tony Hogan. I'd be happy to take any questions.
Gail Besner from Columbus. Very nice presentation. You know, we used to think that portal venous gas was a harbinger or it portended a worse course for these babies. So I might have missed it, but can you just tell us where, you know, portal venous gas came into this? Would it influence your recommendations for the shorter course versus the longer course antibiotics? I'd like to hear more about that. Thank you.
Excellent. Thank you, Dr. Besner, for that excellent question. Portal venous gas was one of the 2 abdominal radiograph criteria that we used for the classification and diagnosis for Bell stage 2. So they were all lumped in together for those who had either or both portal venous gas and pneumatosis intestinalis. As you nicely alluded to, though, I think that's an excellent point that should definitely be considered in future studies for attempting to either risk model or attempt to predict the risk of intestinal stricture formation. As you correctly described, that previous literature, as our previous thinking, was that that may be associated with a higher rate of intestinal stricture formation. But I think more studies and similar work along this line of inquiry might attempt to show that that's not necessarily the case.
Nigel Hall, Southampton. Great study. Thanks for trying to put some evidence behind the duration of treatment for babies with a non-surgical neck. Couple of questions. I'm intrigued why you decided to look at stricture formation as your primary outcome. Do you have any other data, like recurrent neck is one of the reasons that people have cited for using a longer course of antibiotics? And then finally, one of the other benefits of shorter course of medical treatment is you may be able to feed these babies earlier and get them back to full feed and improve their nutritional outcomes. Do you have any data on those?
That's an excellent question. So in regards to your first question with regards to the overall implementation of this protocol, and particularly why we looked at the rate of stricture formation, that was something that we felt was such, particularly for us, a hard radiographic sign that we could point to and then also use in combination with our stakeholders from the NICU to continue with the implementation of this protocol, and then also be able to show them no real worsening radiographic appearance across that. So that was just to kind of as a new study to implement this as a kind of hard clinical and radiographic outcome that we could point to and give them definitive data with regards to that. In regards to your second question, in terms of the initiation of full feeds, we did not specifically record that in this study. Anecdotally, the majority of these patients did receive the reinitiation of feeds as soon as the 7 days of antibiotics were terminated. So that's something that we will continue to look at and would definitely be something important that we're going to include in our manuscript, and something that should continue to be initiated and investigated. But I would suspect that the reinitiation of the full enteral feeds may also play in discovering the true intestinal stricture rate as well.
Thank you.
All right, thank you. Next, we have Arnold Schwarzenegger or Danny DeVito, The Fate of Twins with Necrotizing Enterocolitis in the NICU, presented by Dr. Zane Hellman, Research Fellow at Yale.
Thank you so much for having me. We have no disclosures. Every practicing pediatric surgeon in this room can probably think of a child who succumbed to NEC, who also had a twin sibling who then later went on to develop the disease. Pediatric surgeons have also known what the literature eventually borne out, that children of multiple gestation pregnancies are more likely to develop necrotizing enterocolitis due to generally shorter gestations and smaller birth weights. However, there has been no previous reports on whether a neonate is more likely to develop NEC if a multiple gestation sibling has been diagnosed with NEC previously. We sought to prove empirically what many have known anecdotally. The Pediatric Health Information System was utilized to identify NICU admissions at 47 children's hospitals in the United States between 2017 and 2023, with an admission age less than 28 days. Products of multiple gestation pregnancies were identified using ICD-10 diagnostic codes, and then by matching on date of birth, race, hospital, and de-identified zip codes, we were able to identify twin sets. Next, we looked at ICD-10 diagnostic codes to identify cases of NEC. Once we identified a cohort of children diagnosed with NEC, we looked at outcomes for those children, including using ICD-10 procedure codes to identify the rate of exploratory laparotomy. And finally, we also looked at mortality rate. And because we utilized the NICU admissions from 47 children's hospitals over 7 years, we were able to identify a very large cohort. There were more than 250,000 children admitted to NICUs included in this study, with 11,000 individuals who had a diagnostic code for a multiple gestation pregnancy. Multiples were more likely to be female. They were also had shorter gestations and smaller birth weights. Therefore, it was unsurprising that those children with a diagnostic code for a multiple gestation pregnancy were more likely to develop necrotizing enterocolitis. What was surprising is that those diagnosed with necrotizing enterocolitis who were multiples were less likely than their singleton cohorts who were diagnosed with NEC to require an exploratory laparotomy or die during their primary hospital admission. When we performed a multivariable analysis, we had an unexpected and interesting finding. Those with a diagnostic code for a multiple gestation pregnancy were less likely to be diagnosed with NEC when compared to singleton infants with similar birth weights, gestational age, and other demographic factors as shown here. However, further investigation confirmed what many pediatric surgeons have long suspected. If one member of a multiple set was diagnosed with NEC, there was a significant increase in the risk of developing NEC in the second twin. Similar to reducing birth weight by 1 kilogram and more harmful than reducing gestational age by 1 week. In conclusion, infants born of multiple gestation pregnancies are less likely to develop necrotizing enterocolitis compared to singleton infants admitted to NICUs and controlling for gestational age and birth weight. However, if one infant in a set of multiples develops necrotizing enterocolitis, there's a significant increase in the likelihood that another sibling will develop the disease. We think that these novel insights as found in this retrospective cohort could be due to both genetic microbiomic predisposition as well as infectious exposures. A well-designed prospective trial in the future could be able to tease apart these possibilities and also suggest possible interventions for reducing the morbidity of necrotizing enterocolitis amongst twins. Thank you so much for your time and attention. I'd be happy to answer any questions you might have.
Colton Wayne, Surgical Research Fellow, Nationwide Children's. Great talk. Really like the design of your study. I did have a question. There were a couple points where there was a significant difference in open cardiac surgery, and then as well as in your risk assessment that cardiac defects that were congenital also increased risk. So can you speak to that kind of in the situation of twins and how that may impact neck development? Thanks.
Yeah, so we attempted to control for cardiac outcomes both within twins and the twin sets themselves. And we found that, as would be expected, there was a higher rate of necrotizing enterocolitis among those who had both congenital heart defects and those who had an open cardiac procedure. Because of the way that we conducted the study and not necessarily knowing the timing of which twin developed NEC first, it was harder to control for that when we were looking at the which twin developed based on congenital heart defects.
Ulutoyi Columbus. Really nice study, and I think it's very insightful and helps answer some of the questions we have. But in your data, do you have other data sets that you can actually attach the information you found with birth or pregnancy information? So typically, as you can imagine, twins, even though they're small by the time they're delivered, at that same gestational age, there's a reason they're small, because they're sharing the space, as opposed to a singleton of that same weight obviously has a reason for being much smaller. Now, twins that that both of them have issues, maybe because of some perinatal event, preeclampsia or abruption, that now puts both of them at risk. it'd be nice if you can actually get some perinatal pregnancy-related information to attach to that. I think it'd be very illuminating in terms of what that can show.
I think we couldn't agree more. I think a prospective trial where you're able to kind of both include maternal data as well as the timing of when the infants each develop NEC would be incredibly helpful for further pulling apart this question and having a better understanding of both the disease within twins, but also within singleton infants as well.
Thank you.
Thank you.
Thank you.
Okay, our next presentation is Mechanisms of Intestinal Organoid Transplantation in Enhancing Epithelial Regeneration During Experimental Necrotizing Enterocolitis, presented by Dr. Bo Lee, a postdoctoral researcher at the Hospital for Sick Children.
Yeah, thank you for the opportunity to present my research, and so I don't have to explain about NEC. And so it's multifactorial effects, and so many are affected in the epithelium. So about 10 years ago, and so we actually using organoids treated DSS-induced colitis experimentally. And so basically 3 years ago, and so there has been first clinical trials using this method. So the aim of my study is actually to doing the same thing and using the organoids transplantation to see whether they have the effects on the experimental NEC model. So the experimental design is like we're taking the GIP organoids, and so we dissociate and can transplant it by oral gavage and also the animal into the GI tract. And so only by one dose at P6 during the NEC induction from P5 to P9. So we trace them like 3 weeks later and also 3 months later. And so this is the cartoon, and so we designed, and so basically we give it from the mouse, and also we from the other side. And hopefully they all go to the small intestines. So first we observed is the organoid transplantation is OT. They can improve the survival of the experimental NEC. And secondly, and so for taking the morphology, and so we do the HE staining and also the histological scores, and so the OT can reverse the intestine damage. And also the INOS, which is inflammation markers, and also the LGR5, which is the markers for the stem cells. And so because we give the GIP organoids, we can trace them where it goes. So basically it goes to the small intestine, where is the most affected in NEC. And so regardless of which way you go from the top or you go from the bottom. And also they go to the bottom of the crypts, which is the green dots at the bottom of the crypts. However, as 3 weeks later or 3 months, and so we never see any regeneration intestines from these GIP mice or GIP organoids. So which is not the same as we observed in the DSS colitis model. So they basically can regenerate the gut. So which is you see from the red at the bottom of the corners. So to understand the mechanism of that, and so we're doing the single cell analysis and see whether they have some indicator and how they works. And so we only focus on epithelium. And so we see there's a switch in NEC, and so basically enterocyte is reduced, and so they try to tend to be like more on the goblet cells and to build up more barrier functions and protect. So that's in NEC. So after OT, and so in the OT, they can reverse this phenomenon. So they can get more enterocyte, and so they can also keep the goblet cells as some high level expressions. So basically they're pushing more enterocyte as well. Taking more deeply and to do the stem cell analysis, and we take the stem cell groups out, and so we actually identify these like number 8 subclusters, so which is unique subclusters, and in the NEC, which is the disease is exclusively associated. And so we are doing more research on these groups of cells, and hopefully these could be the ones, and so we can reverse, we can target and to reverse the phenotype. And also we did some bulk RNA analysis, and so there's many pathways and being upregulated, downregulated. So I want to give one example, which is the first one, the C4 pathways. So it's related to the mucosal immune response. And so they release the antimicrobial peptide, and so which is related to the Panny cells. And so you can see from the Malin studies, and so Panny cells actually is quite dramatically increased after OT transplantation. So what is the Panny cells? Panny cells is expressed at the bottom of the crypts and can release wind and can support the LGR5, which is the stem cell niche. However, in the early stage of the newborn babies or newborn pups, so these cell type populations is absent. They are not mature enough. So we did some staining, confirmed the story, and so basically after the organoid transplantations, we can see a huge increase of the mature Panny cell markers, lysosome 1, at the bottom of the crypts. So the conclusion of our study is we can use intestinal organoids as a treatment strategies. They can repairing the intestinal epithelium damage, so they can intercrypt to the bottom of the crypts, and but they do not regenerate the gut. And so the mechanism of actions can be stimulate the maturation of the Panny cells and maintain the stem cell niche. And so finally, I would like to thank you for the lab, and also my family is there. Thank you.
Gail Besner from Columbus. I've been really excited about this work since I heard your group present this, quite a while ago now. So congratulations. Can you tell us a little bit more? I may have missed it. What is the origin of the crypts? Are these from induced pluripotent stem cells? Are these primarily derived from epithelium, from humans? Just tell us more about that. How long do you culture the crypts before you transplant them? And tell us more about why do you rupture the crypts before you administer them? Thanks for a great talk.
Okay, thank you. Yeah, so that's all the great questions. And so we actually have, it's been a long time to develop this project. And so we do have a lot of data regarding that. And so we're actually using different origins, as you mentioned. We use the small intestine organoids, we use the colon organoids, and also we try to use tissue derived, which is because we are the surgeon lab, and so we have the tissue access. And so we have access to the IPS derived, but we never tried that before. And so compare those two resources, and so it could be interesting as well. So that's one thing. And also we try is like the whole organoid transplantation, and also the crypts, the break it down crypts transplantations. So we get the same effects. And so the take-home message is like, the organoids or the cells can go in there, but they do not repopulate. And so they send the signals or they send some factors there. We are more like focused on which factors actually released by these organoids or by these crypts. So that's the directions. So we are deep more. And so hopefully the paper is published like soon. And so we are wrapping up of this project, as you said, and it's already a long time. And so it's nice to wrap it up. Thank you.
All right, our last presenter for the day is Dr. George Bios. Okay, sorry. Research fellow at the Hospital for Sick Children. Changes in glial activity or activation states in pyramidal region of the hippocampus during necrotizing enterocolitis.
All right, thank you for the opportunity to give this talk today and talk about the brain. So about 45% of NEC survivors suffer from neurodevelopmental impairment. And this is impairment in their cognitive, psychomotor, and mental development. A lot of clinical investigations have found structural abnormalities that aren't related to prematurity alone. So the early neonatal life, as we know, is a critical developmental window both for the intestine and brain. And when we look closer to the brain, there's a lot of processes that are ongoing, including myelination, synaptogenesis, so a lot of maturation processes. So we really wanted to understand how different regions of the brain are vulnerable to stressors, specifically to the stressors we get in NEC. And when we focused in on one region called the hippocampus, which is the region that's central to memory formation and coding, it's becoming known that it's one of the most susceptible areas of the brain to injury during early neonatal life. And the reason is it's undergoing a huge growth spur at this time, and it's marked by increased connectivity, neurogenesis, and in particular, the lack of vascularization and the neuron-dense nature of this region underlies vulnerability to stressors. A lot of research in the last few years from Dr. Hawkins' group and others have found the mechanism of brain injury during NEC, and it involves bacterial ligands and monocytes that circulate to the brain past the blood-brain barrier and activate TLR4 signals in the macrophages of the brain, called the microglia. And this initiates a neuroinflammation phenotype, and it really affects the maturation. So we hypothesized that, in particular, the hippocampus is one of the first regions to be injured, and how we define hippocampal development is in two ways. We define it by the architecture, which is how the cells are organized and distributed, which is important for their function, and also how the cells communicate with each other in the cell-to-cell signaling network. So we think that NEC impedes the hippocampal architecture and cell-to-cell signaling. And so for this study, we wanted to look at more closely to the glial cell type. So this is the microglia and astrocytes that sort of survey injury. And we wanted to look at the transcriptomic changes within the different hippocampal subregions. And as we heard today, we used the neonatal mouse model of NEC, whereby it's induced by three factors: the hypoxia, the hyperosmolar feeding of formula, as well as gavage feeding of LPS, and we confirmed NEC with the histological grading and the gene expression of pro-inflammatory markers. So our approach was to use spatial transcriptomics, which allows for single-cell analysis with spatial information, and we used the MERFISH platform, whereby we took sections of the brain. We outlined the hippocampus anatomically, and we performed single-cell analysis where we identified the different cell types, and we did some gene expression analysis. So within the neonatal hippocampus, there was a variety of non-neuronal cell types. So the major cell types were the oligodendrocytes, or the myelinating cells. There was a variety of astrocyte cell types related to both progenitor and also in more mature cell types, vascular cell types, and as well in the macrophages, we identified both the resident microglia and also non-parenchymal macrophages. And so when we identified these cell types, we plotted them spatially, and this is the hippocampus. And when you focus in on the blue cells and the orange cells, so these are the microglia, and they're surrounding the neuronal green cells, these are the pyramidal neurons that you see, and in the dentate gyrus, you see the purple cells, which are the granular cells, so the progenitor cells. And what we found was that in the NEC group, the astrocytes really lacked their maturation markers. So we identified a lot of sort of canonical markers of maturation and differentiation that were significantly reduced compared to their best bet control. And when we did enrichment analysis, we identified key regulators of Wnt signaling, and as well as cytokine signaling, and more interestingly, VEGF signaling and regulation that was reduced, which are all critical for maturation of the astrocytes and also the cells they support, the neuronal cells. And on the other hand, with the macrophages, specifically the microglia and non-parenchymal macrophages, they lacked homeostatic markers. And those homeostatic processes, also including VEGF and cytokine signaling and Wnt signaling regulation, were also dysregulated and reduced in gene expression. So in conclusion, the severity of early life hippocampal development alterations may underlie the NEC-associated neurodevelopmental impairment. And NEC alters the phenotype of glial cell types, indicating response to injury in the hippocampus. So what are the implications and translational considerations here? So with current strategies, we could think that timely excision of the necrotic intestine early on can protect the brain from the damage. And from the studies we're conducting now with transcriptomics, we can really go into the field of precision medicine and find targets within these disease cell types that we could possibly target with pharmacological means in vulnerable brain regions such as the hippocampus. Thank you to my lab, Dr. Piero, and our funding agencies, and thank you for your attention. I'll take any questions.
Hi, Kelly Austin from Pittsburgh. That's really interesting, but I also didn't really catch, how did you control for just the generalized hypoxia? Did you test this in just other models of NEC that didn't have systemic hypoxia, or?
Yeah, that's a great question. So when we looked at the experimental model, we did break down the model into its individual factors. So the LPS alone, the hypoxia alone, and also the gavage feeding alone. And we compared it to all of those in combination, specifically with the administration we do in the NEC model. None of those factors alone, specifically also hypoxia, were giving us NEC, and also the injury we see in the neck brain, experimentally, yeah.
Did you test the glial cells in the bowel?
In the bowel. So we have done investigations with you mean the macrophages? Or the,
I meant more of the enteric.
enteric glia, yeah. So specifically, we've investigated, and also Dr. Hawkins' group has done a lot of work, and the TLR4 signaling also does play a role in the enteric glia in the intestine, which affects motility. It's part of the pathogenesis of intestinal dysmotility in neck, yeah.
Hi, Helen Shea from SUNY Stony Brook. Lovely work. It's a beautiful map. I have a quick question about the oligodendrocyte precursor cells. How do you predict that will affect the expression or the expression pattern of oligodendrocytes later on? And are there migratory patterns that you would predict? Because this is during the fourth wave of migration of your OPCs.
Yeah, this is a great question. I didn't present this in the study, but demyelination is a hallmark of NEC-induced brain injury. And we're learning that the oligodendrocyte differentiation processes from their progenitor cells is affected. So what we're looking at now is understanding which markers are affecting first of all, their cell death, their viability, as well as how they're maturing. So we're doing a deep dive into the trajectory of the OPCs via the single cell analysis. So my colleague earlier did a melanin analysis, which is a technique, computational way we can identify the trajectory of immature to mature cells. And so that's our sort of next step. And I predict what the migration patterns are affected. So for example, I showed you the granular cells, which have tangential migration of the neural precursors, and which over time they mature into granular neurons. And we do see with the architecture effects there, and also the signaling, that tangential migration is affected. So we expect that the oligodendrocyte progenitor cells have a similar change in the architecture and their organization. We're looking further into that as well, yeah.
Thanks, Chad. All right, we're going to be finishing a little early, leading out into the afternoon break, and the next scientific sessions start at 4. Afterwards, 6 p.m. is the cocktail reception, followed by dinner.